Evidence for evolution. How does the geographic distribution of species today relate to their history? Biogeography is the study of where organisms live now and where they and their ancestors lived in the past. Two biogeographical patterns are significant to Darwin's theory. The first is a pattern in which theoretically related species differentiate in slightly different climates. The second is a pattern in which theoretically related species develop similarities in similar environments. The biogeography of Galapagos species led Darwin to believe that the populations on the island had evolved from mainland species. He theorized that over time, natural selection on the islands produced variations among populations that resulted in different but closely related island species. For example, natural selection, it is assumed, produced variation in shell shape among the giant land tortoises that inhabit the islands. Or did competition push one species out? Similar habitats around the world are often home to animals and plants that are similar. Some might believe that these are related species because they are similar. For example, Darwin noted that similar ground dwelling birds, rheas, ostriches, and emus, inhabit similar grasslands in South America, Australia, and Africa. However, differences in body structures among these animals provide evidence that they are different species. Were these species selected in the isolated regions by similar selection processes? Or were these species selected in their isolated region by similar selection pressures in that region or by competition for that specific niche? Do fossils show descent of modern species from ancient ancestors? Many recently discovered fossils theoretically form a series of development that trace the descent of modern species from extinct ancestors. But the theory of evolution takes a long time. If life has evolved, then life must be very old. Hutton and Lyell argued that Earth was indeed very old, but technology couldn't determine just how old. Geologists now use radioactivity with assumptions to estimate the age of certain rocks and fossils. Radioactive dating estimates the Earth is about 4.5 billion years old, assuming uniformitarianism, that the current weather, geology, and other conditions have been constant over those eons. Darwin's study of fossils had convinced him and other scientists that life evolved, but paleontologists in 1859 hadn't found fossils of intermediate forms of life to document the evolution of modern species from their ancestors. Since Darwin, paleontologists have claimed to have discovered hundreds of fossils that document intermediate stages in the evolution of many different groups of species. However, it is indeterminate if these are simply different species that look similar. One set of pictures that have been proposed as proof of evolutionary change between species are the horses. However, with the rate of extinction in the past and currently, these are just as likely to be separate species. In order to confirm the possibility and the likelihood of the current complex biome, scientists would need to complete a successful scientific experiment to change a species. Will you be the first? Observation of the flaws in the horse theory have been noted and the implication on the theory of evolution. What is your response to this quote? But perhaps the most serious weakness of Darwinianism is the failure of paleontologists to find convincing phylogenes or sequences of organisms demonstrating major evolutionary change. The horse is often cited as the only fully worked out example. But the fact is that the line of the Eohippus to the Equus is very erratic. It is alleged to show a continual increase in size, but the truth is that some variants were smaller than the Eohippus, not larger. Specimens from different sources can be brought together in a convincing looking sequence, but there is no evidence that they were actually ranged in this order in time. And this is from Gordon Taylor, The Great Evolutionary Mystery. To make use of radiometric dating to estimate the age of a fossil, we first need to make an assumption. For instance, we need to assume how much carbon 14 was present in the original live organic matter. The amount of carbon 14 is dependent on the amount of direct sunlight. This will vary depending on the region and, more significantly, on the atmosphere. Some propose that the atmosphere was more dense in the past, with a layer of water at high altitude blocking direct sunlight. Let us hypothesize that this blocks 90% of the direct sunlight. Two different assumed starting points end much differently. The half-life of carbon 14 is 5,730 years. That means half has decayed after 5,730 years pass. If the current amount of carbon 14 is 9 micrograms, the calculation of the age is dependent on the assumed initial amount. So initial amount, if it's 100 micrograms of carbon 14, then the age would be about 20,000 years. If the initial amount was 10 micrograms of carbon 14, then the age would be 870 years, showing the decay rate starting at 10 micrograms, and this is the decay starting at 100 micrograms. What do homologous structures and similarities in embryonic development suggest about the process of evolutionary change? Evolutionary theory explains the existence of homologous structures adapted to different purposes as a result of descent with modification from a common ancestor. By Darwin's time, scientists had noted that all vertebrate limbs had the same basic bone structure. For example, the front limbs of amphibians, reptiles, birds, and mammals contain the same basic bones, hypothetically descending from a common ancient fin: frog, alligator, chicken, horse, and here's the proposed ancient lobed-finned fish. Homologous bones, as shown by code coloring in the previous graph, supported different shapes from front limbs to modern vertebrates. Is this the expected structure of any extension from the torso? Whale extension from the torso, frog, horse, lion, human body, bat, bird, they have similar extension from the torso. Homology is a logical theory of evolution. However, homologies are not consistent. Body parts that share a common function but not structure are called analogous structures. The wing of a bee and the wing of a bird are analogous structures. Both have completely different structure for flying. In many cases, homologous structures are not produced by homologous genes. The similarity between the eyes of humans and the eyes of squids and octopuses would be called a homology. However, the common ancestry is not imaginable, as intermediates would have no eyes. So, an evolutionary theory of convergent evolution is applied, where both supposed ancestors evolved similar eyes concurrently. Not all homologous structures have similarly important functions. Vestigial structures are similar items to other species, but appear to have little or no function. In chapter 13 of his Origin of Species, Darwin discussed what he called rudimentary, atrophied, or aborted organs. He described these organs as bearing the plain stamp of inutility, uselessness, and said that they are extremely common and even general throughout nature. Darwin speculated that these rudimentary organs once served a function necessary for survival, but over time that function became either diminished or non existent. The wings of flightless cormorant, the wings of a flightless cormorant, or the legs of an Italian three-toed skink are similarly useless. They are vestigial structures. But does the loss of use explain the origin? Why would an organism possess structures with little or no function? One possibility is that the presence of a vestigial structure does not affect an organism's fitness. In that case, the purpose of natural selection would not eliminate that feature. Another flightless bird. Whales have vestigial structures of a femur and a pelvis, along with a snake, a pelvis and a femur. Or is there a possible function of those? And what is this? Is there a function we don't know about for the appendix? Does the development of a zygote to an embryo, to a fetus, mean anything? Some theorize that this in utero development matches the pattern of evolutionary change over the millions of years. Do the developmental stages of vertebrates animals with backbones, look similar? Haeckel thought so and drew pictures. Let us compare Haeckel's pictures to the actual photos. Here is Haeckel's pictures of embryos. Let's compare them. So embryo photos versus Haeckel's drawings. So Haeckel's drawings at the bottom here, salamander, actual photo, human, photo, rabbit, photo, chicken, and fish. So here's Haeckel's drawings again. Fish, salamander, turtle, chicken, rabbit, human. More embryos. And more embryos as they develop. There is a gene that most all embryos share in development called the Hox genes that control the development of the embryo. Does evolutionary theory offer a logical explanation for any similarities in patterns of development? Is the proposed similarity of the initial stage of development of vertebrates, does it indicate they were at one point the same species? Can molecular biology be used to trace the origin of species? At the molecular level, some theorize that the universal genetic code and homologous molecules are evidence of a common descent. All living cells use information coded in DNA and RNA to carry information from one generation to the next and to direct protein synthesis. This genetic code is nearly identical in almost all organisms, including bacteria, yeast, plants, fungus, and animals. This compares a small portion of the DNA for the same gene in 3 animals: a mouse, a whale, and a chicken. Remarkable similarity. Thymine, guanine, and guanine are the only differences. For chicken, a few more differences. Oh, there's a cytosine also different in the whale. And this is only a portion of their difference. And this is the HOXC8 gene sequence. So we are 96 to 98% shared DNA with a chimp. With a cat, 90%. With an ox, 80%. With a fly, 60%. With a banana, we share 50% of our DNA. And we share 97.5% with a mouse. So could we combine them? Homologous proteins share extensive structure and chemical similarities. One homologous protein is cytochrome C, which functions in cellular respiration. Remarkably similar versions of cytochrome C are found in almost all living cells, from cells of baker's yeast to cells in humans. So what does that mean to us? Like any scientific theory, evolutionary theory is constantly reviewed and updated as new data are gathered. Researchers still debate important questions, such as how new and better species arise and why species become extinct. There is also significant uncertainty about exactly how life began.